So now let's talk about the polysaccharides. And if you remember, the beginning of the class, we talked about that the carbohydrate has two main functions. One as a fuel and the other one as a structural. Yeah, so we're going to show you examples of that. Here we have three polysaccharides, one in the potatoes, two in the human, that is whole in the potato, and three in the plants. And some of them will be used, the first and the second will be used as a fuel, and the third one, it will be used for a structure of the plants. So let's talk a little bit about these three. So in the first one, we have the starch in the potato. And if you see, here you have all these are glucose, are monomers. And but they are not completely straight. They are bending. And why they bend? Because of the type of glycosidic linkage. So here you have one starch is used for as a fuel. When you break or the potatoes are hungry, you just bring this and you have a glucose. So here you're using this for fuel, but in this case specific for storage of the fuel. In humans, we don't have a starch, we have glycogen. And the glycogen, again, if you see the bending, here they also bend, they have some branches because of the way that the glycosidic linkage work, and we will talk in a second more about that. So while in plants they have starch, in vertebrates we have glycogen, but they have the same function. When we are hungry, we just break this glycosidic linkage. We bring this polymer and we release the sugar to use it as a fuel. Finally, the last polysaccharide that we're going to talk is called cellulose. And in the cellulose, if you see, it's very different. It's very organized. Yeah. And if you see, it forms filaments. And is used as a framework in the cells. So to summarize, I talk about the starch that is for storage. I talk about glycogen that is also for storage. And I talk about cellulose that is for structural. So polysaccharides, the polymers of sugars, have have storage and structural roles. And in the next slide, we will see why they look different, why some of them branch, while other ones are linear. Now, polysaccharides can be divided by two parameters. The first one, the position of the glycosidic linkage, and I will talk more about that in a second. And the other one, for the type of monomer that they have, if it's a glucose or is a fructose. Let's dig into that. So now let's see the difference between this alpha glucose and this beta glucose. And I want that you stop the video and try to see if you see what is the difference. Okay, if you stop the video and you detect, here is the difference. And here is the difference. So while in the alpha glucose, the OH are close to each other in the carbon 1, in the carbon 1 in the beta glucose, the OH are separated, are facing in different directions. And if you see the starch and the cellulose, here this is the starch, and if you see, it's very similar to the cellulose. Both use the same linkage, both use monomer of glucose. However, the starch will be alpha, and the cellulose use a beta glucose. So, as I said, like starch, cellulose is a polymer of glucose, but the glycosidic links differ, that one is alpha and the other one is beta. Now let's talk about starch. And we know that it's an starch polysaccharide. And maybe you have seen this picture in high school. You remember what is a chloroplast. It's an organelle that is in charge of the photosynthesis. So photosynthesis take place in this in chloroplasts and in our plastids, and as they take CO2, light, and water, and they turn it into sugar and oxygen. So they need to store all this glucose, and they store it in form of a carbohydrate called starch. And there are two main components of the starch, one that is the amylose and the other one that is the amylopectin. And they're very similar, both have alpha glucose monomers, but the difference is in the type of glycosidic linkage. One is 1 to 4 and the other one is between the carbon 1 to 6. So to summarize, starch is a starch polysaccharide of plants and consists entirely of an alpha glucose monomers. In addition, plants store the surplus of starch as granules within the chloroplast and or plastids, and the simplest form of starch is the amylose, and although they have another one called amylopectin, and the only difference is the type of glycosidic linkage. One is between the carbon 1 and 4 and the other one is between the carbon 1 and 6. Okay, now let's talk about another starch polysaccharide. And this starch polysaccharide is located in animals, fungi, and bacteria. And the name is glycogen. And glycogen is the monomer is the alpha glucose. And you remember that the starch was located in the chloroplast. So in the case of the animals, they don't have chloroplast. So this one will be located in the mitochondria. Here you can see some granules of the glycogen. So you have all this surplus of alpha glucose, and what you do is to storage in granules in the mitochondria. And remember, what part of the body is the one that needs more energy? The muscles. So in the muscles and also in the liver, you have a lot of glycogen reserves in case that the body needs them. Now, something that is important is that if you see, although they have an alpha glucose ring, the glycogen has two types of glycosidic linkage. One that goes to the carbons 1 and 4, and they form the ring like that, and the other ones they form the ring joining the carbon 1 and 6. So, glycogen is an storage polysaccharide in animals. Also, glycogen has alpha glucose rings. And finally, human and or vertebrates store glycogen mainly in the liver and in muscle cells. Now I'm going to talk about a structural polysaccharide. I'm going to focus on the cellulose. And the first thing that I want that you remember is that the cellulose is a major component of the wall of plant cells. And as this schematic shows, the cellulose has a beta glucose like here. And another important thing is that if you see these fibers that are in parallel, all this parallel structure allow them to form a strong building material for plants. And this is why they are located in the cell wall. In addition, something important about the cell wall is like herbivores, like cows or termites. they require the interaction with microbes to break into small pieces to turn these polysaccharides into monomers that they can be digested for the cows. So herbivores, they have a symbiotic relationship with these microbes to be able to digest this type of food. And finally, we're going to talk about another type of polysaccharide that we haven't talked before, and that is located in the exoskeleton of insects and also in the fungi. And this is the structure of the chitin monomer. So if you see, this has this nitrogen here that is very atypical. It has a glucose that is a beta, and is the glycosidic linkage is between the carbons 1 and 4. Very interestingly, this material seems to be has applications in medicine because it's flexible, but at the same time is very durable. So it can be used during surgeries, and later it decay without need to be remove the stitches. So chitin is another structural polysaccharide and is found mainly in the exoskeleton of arthropods, and also provide a structural support for cell walls of many fungi. And as I said, chitin is used as to make a strong and flexible surgical thread that decomposes after the wound or incision heals. So this is a pretty cool type of application, and just for you to remember, something that sometimes appears in the exams is that this having nitrogen, something very atypical, and is the only polysaccharides that we have talked today that have nitrogen. To summarize all the things that we have talked, the polysaccharides are polymers of sugar that have two main functions. They can be for storage, or they can be for structural roles. And I think this chart summarizes very well all the things that we have studied. They can tell you the source. So the cellulose is located in plants, and as well as the starch, while the glycogen is an animal, located in animal. The subunit here, the only one that we studied that it was beta glucose, it was the cellulose, although I talk a little bit also about the chitin that is not included here. While the other ones are alpha glucose, and if you see, the cellulose have 1 to 4 glycosidic linkage, while in the amylopectin and the glycogen you have these two types. In the cellulose there is no branches, so this is what they look, they shape like this. And remember, they are parallel because they form this parallel structure, this microfiber that can be very useful as a building material in plants. Well, look, the amylose have coiled structure, and the amylopectin and the glycogen have this branched structure. So I think hopefully this chart can help you to summarize all the things that we learned today. Have a good day, and if you have any questions, just let me know.